and the John and Patricia Klingenstein Fund in honor of Daniel Callahan, a very important American philosopher and co-founder of the Hastings Center. I'd like to express my deep appreciation to the Klingenstein family for their vision and support. I also want to say how much we appreciate mounting today's event in collaboration with the Center for ELSI Research and Analysis, the acronym SARA. This is a project that's led at Columbia University by Dr. Sandra Sujin Lee, who heads the Division of Ethics at the Department of Medical Humanities and Ethics at Columbia. From its inception, the Hastings Center has had two equally important missions. We're a nonprofit, nonpartisan bioethics research institute producing scholarly analysis on a wide range of ethical questions in health, healthcare, and the life sciences. We publish reports, peer-reviewed articles, produce congressional briefings, and own two journals where current issues in bioethics are debated among experts worldwide. In other words, we're a think tank, but we're a think tank with a difference. The Hastings Center has a co-equal commitment to public engagement. We believe that in a democracy, educated citizens should participate in decisions raised by advances in biomedical innovation. They should know about and be able to consider policies to ensure fair access to the benefits of new technologies and to establish guideposts capable of mitigating potential harms. And it's journalists who are absolutely essential to informed public engagement. And yet, as a profession, you are under increasing assault. Business models for journalism have changed dramatically, creating really big challenges. And now the proliferation of fake news has made it extremely difficult to figure out how to help people distinguish truth from illusion. So, I have enormous respect for journalists. Our democracy depends on you, and bioethics needs you too. We've launched this series to support you in the work you do. We hope you'll find that the speakers we bring to you and the conversations we stimulate help you identify the ethical issues raised in health and science and identify them and then investigate them more comprehensively. The 2021 series, meaning everything we're going to do between now and December 31st, is called Genomics in Society, New Developments, New Questions. And this series is going to focus on the implications of new research in genomics. Today's event specifically is on new research on genomic influences on traits like intelligence and educational attainment, an area of study that is rife with promise, but also with the potential for misunderstanding. So it really needs you to help the public understand and enter this space. It's my pleasure to introduce our distinguished moderator, Amy Harmon. She is a two-time Pulitzer Prize winning journalist for the New York Times. Amy covers the intersection of science and society. The Pulitzer Prizes she won were for The DNA Age, which examined ethical issues with DNA testing, and also for a series in which she was a member of a team of reporters called How Race is Lived in America. Amy, thank you so much for agreeing to moderate today. We really appreciate your participation and your leadership. And now it's time for me to pass the baton to you.
Thank you so much, Millie, for that introduction. And thank you everyone for coming. I realized yesterday when I was getting ready to kind of send the usual promotional tweet to like remind people that this was happening, and I was going to say, I was looking forward to this panel. Like that's kind of what one says, right? And I realized that I couldn't exactly say that I was looking forward to it because there was this part of me that was kind of dreading it. And that's not a comment on the panel itself, because we have the best possible panelists for this event, which is titled Genomics, Human Behavior and Social Outcomes, a discussion for journalists. And I don't think it's because I'm like the type to shrink from controversial topics. I think that this discomfort that I feel stems from my own sense that I don't know journalistically the best or most responsible way to approach covering some of the issues that are growing out of this newish field called sociogenomics. And I feel like if you asked me how I would approach covering genetically modified food, or the politics of the COVID vaccine rollout. I mean, these are topics that like I have an opinion on how to do that, and this is hard, and the stakes are really high. So I'm just gonna, I was asked to give a few like framing remarks, so I'm gonna just give very brief framing remarks, and then I will introduce the amazing panel, and then we will start to have a conversation that I really, I just think really needs to be started. And I'm really glad that we're here to do that today. Yeah, so the stakes are high. So I want to say something about that, because I mean, we were talking about genomic influences on human traits that, at least in our society as it is currently constructed, really matter, like really make a difference in terms of, the amount of wealth or status or success or, possibly even happiness that people are likely to accrue. And we're talking about educational attainment, how far you're going to go in school, intelligence, sexual behavior, risk taking, these types of things that are, and they are also, they are the same traits that have been used to, in the past, justify eugenics, forced sterilization, genocide, like the worst atrocities, and right. So that really weighs on me when I am thinking about how to approach talking to people and framing a story about this. And these are also the kinds of traits or ideas or stories that will make your editor's eyes light up, right? So like, and we there's a lot of pressure on us to make our editors' eyes light up, and so it also is another reason we have to be really kind of careful about it. And I guess just to say a few words about the why now, like why are we talking about this now? I think maybe a lot of people in the audience are journalists who already know this, but many years ago, geneticists realized that there was not going to be a gene for a particular trait, that pretty much every complex behavioral trait and most medical disease predisposition traits are going to be polygenic, right? The weighted sum of thousands of variants throughout the genome, and that would require a lot of genomes to try to get at what those variants are. And so now there are a lot of genomes, and the same databases of genomes like the UK Biobank and 23andMe that are being used to query to try to sort of home in on someone's risk of heart disease are being used to generate polygenic risk scores that are related to, as we've said, and our panelists will describe more, what it means to be educational attainment, how far you're going to go in school. And I guess I see at least two, maybe three, key journalistic challenges to this, and one of them is that there is a lot of dissent within the field itself, within sociogenomics, and geneticists who are population geneticists and sociogenomicists, the people who work with this data, I think are not settled within themselves on how the data should be parsed, when it's being parsed reliably. And we'll just give one example of that, which is so Paige Harden, who is a psychologist at the University of Texas, has just written a book, and many of us may have read the excellent Gideon Lewis Krauss profile of her in the New Yorker a few weeks ago. So she's written a book which I do think everyone should read who's planning on covering this, called The Genetic Lottery. And she makes a really impassioned case for why polygenic risk scores for like educational attainment in particular should be used to sort of think about how society is structured unequally, that genetic luck, as she calls it, should be factored into luck, just like parents. We all sort of are more familiar with thinking that, well, it's not your parents' wealth is not, you didn't earn it, like you're to some degree lucky, and that we might think about redistributing wealth based or resources based on that, and that she's sort of arguing that we should also take genetics into account now that we can measure it more precisely. But then there will be people who, I mean, if anybody has followed, there's just been some discussion of Paige's book on Twitter in the last few weeks, and another sociogenomicist at University of Wisconsin at Madison, Jason Fletcher, who has done work with this himself, jumped in and sort of challenged the idea that, whether should you really look at the genome-wide association study results themselves as representative, or can you really only count on the between sibling, the within family GWAS results? And I mean, it begins to sound technical, but there's really a lot of debate about that issue. And so as journalists, I think we're maybe used to being able to call, you write about a study, you interview the author, you call up someone who's familiar with the work but wasn't involved in the study, and they they give you kind of like the what the field thinks about this. And usually it's like, yeah, this is a really this is a really important study because it was on the cover of Nature or whatever. So it's not that you don't have to decide that much. I mean, there may be people with different views, and you try to find someone with a more critical view also. But it's I feel like in this case it's less settled, and that's just one. And that's sort of, now I was just talking about sort of within population disputes about how to parse GWASs that are taking place within a particular ancestry group. There's another important divide that we will speak to. We're lucky to have a population geneticist also on our panel, but it's really about how to whether these results can be informative about different populations, ancestral populations, so people of European descent versus people of African descent. That obviously it's like it gets into all the super touchy issues about race, but also more properly ancestry. And there's anyway, we will get into it in the panel, but I'm just trying to sort of outline some of the that's another challenge for us as journalists to have to sort of figure out what some people are saying. Oh, this doesn't really apply to between population. We don't have to think about that. We don't have to look at the different, how these results look differently in different populations, and others are not saying that. So okay, and then the second challenge I think for us is like, let's say there is some thing that is really, we need to be writing about this, and we need to be the science, there is some degree to which these results are real and matter, and so then it's sort of like, and this is where Eric comes in. Should we be also interviewing ethicists when we write our stories about our sort of straight science study stories? How do we this is not about like what drug can best, how your heart disease predisposition can be better predicted by these results. This is about how far are you likely to go in school. And that's kind of, I mean, the idea of people having scores for that, or even groups of people having scores for that, is more, Paige Harden would argue, is important and encouraging for potential social equality issues, but it's also really creepy. So there is a third challenge, and I'm almost done, almost gonna hand it over to the panelists, but I guess I would just say, so I dipped my toe into this a few 2 or 3 years ago now. Everything pre-pandemic is a blur, but I wrote a story when the alt-right was ascendant, about how white supremacists were kind of taking research results from human population genetics and some of these sociogenomics results, and sort of distorting them and extrapolating from them, and trying, as has been done throughout history, to use them to sort of justify their views of racial hierarchy. And one of my frustrations with that story was getting geneticists to talk about it, like, I mean, on the record. And so because it's so touchy, and because it's hard, and I came to understand why it's hard. There's not like always the silver bullet answer to this, but I do think that's changing. And so my story actually ended up being partly about like why human geneticists were not really talking about this stuff. But I think that is changing, and so we have this panel here where people are going to be talking about it. I wanted to just end my introduction remarks with to give you a sense of of the stakes and how even the scientists. So for this panel, people were registering, and it was said that it was, I think the Hastings Institute made it clear that this was for journalists, but apparently lots of scientists also wanted to sign up. So geneticists were signing up, or they were contacting me asking me if they could get in, even though they weren't technically supposed to be in the audience. And we did limit it to journalists, although I guess anyone can watch the video. But one of them wrote to me a question, and so this is from Robbie Wodeau, who's a postdoc at the Broad Institute and who is an author on several of, there are many authors, an author on the same-sex sexual behavior study, and that will come up in our panel, I think, and also the educational attainment study and some others. And he just wrote to me, because I told him, you're not going to be you can't be in the audience, but he said, but one thing I'd really like to know is, he wrote, so it is very clear that the type of work that I and others do has the potential to hurt people if the communication or outreach is wrong, or even if it's right. So should we be doing this work at all? As you know, even though I researched this stuff, I'm not totally sure. I'm very curious about what others think as well. Like you said in your post, that was referring to my tweet, the stakes are very high here. So I don't know that we will answer that question in this panel, but I just sort of use that as a starting point. And now I'm going to hand it over to these panelists, which are, so Melinda Mills, who is a sociologist at Oxford and who does sociogenomics, and Arba Harbeck, who is a population geneticist at the University of Texas, and Eric Perrins, who is a senior research scholar at Hastings. And they will each talk for about 10 minutes about sort of their relationship to this topic and the questions that concern them. And we'll have some discussion and we'll have your questions. Thanks. Handing over to you, Melinda.
Thanks. Thank you very much, Amy. And it's nice to see everybody. Well, not see everybody, but at least to address you in relation to this. So I think it's good. Amy's given an introduction to sort of tell you about what sociogenomics is. And I think what it is really introducing ideas from molecular genetics into social behavioral sciences. So I'm in demography and sociology. And so the things we study are often educational attainment, income, well-being, and I study reproduction. So when you have children and your first child. And so they're usually what's called complex behavioral outcomes, but there's a lot of medical outcomes as well that are complex, such as BMI or type 2 diabetes or other traits like that. So, and as Amy said, I don't need to go through and explain what a polygenic score is, so it's just many different genetic contributions, and I'm sure that will come up because that has been the large discussion. So why am I doing this, and why are sociogenomics, why are we doing this in the first place? And I think, you had the GWAS study. So these are genome-wide association searches. And you can think about them as genetic discoveries where you search across the genome and we find many so many different genetic variants that are associated, so correlated, related to the outcomes that we're studying. And then I think as of 2019, you just had this explosion in types of data and in computational possibilities and statistical techniques to study it. And I come from a background of demography, of studying fertility. So when you have your children, how many children you have, and sexual behavior, what ages, and all of these different aspects. And so for me, I was looking at things largely from a socially deterministic lens. So everything was related to, people are having children later or earlier in relation to their social environment, in relation to childcare, contraception, work-life reconciliation. And it became increasingly clear that, as individuals shifted, having children till later, they were having infertility problems. And so something, could have been happening in relation to genetics related to infertility or endometriosis. So there's other traits, that could have resulted in these people being infertile. And then on the other side, we had teenage pregnancies, in certain areas and groups. And we found a study we published in just a few months ago in Nature Human Behavior. It was related to, addictive traits, risk, self-control genetically. So I was interested from it, studying decades on this topic from the social sciences to look, well, what are the biological components? So it came from that side. And I think when we talk about these polygenic scores, these multiple genetic variants, I think it's really important, and Amy already discussed, I think there's some disagreement definitely within the sociogenomics community, about these things. And it's not just, I don't think it's just us, I think it's in the general, genetics community. So, often many of us don't think of it as an individual predictor as it is supposed in some books and by some people. Robert Ploughman, who wrote Blueprint, famously talked about it as, we can have precision education policy, and we can reflect on that later. But there's a few problems with that, because if you look at the polygenic score for educational attainment, and you do a scatter plot, of educational attainment by years of education, you'll see, if you get into the top 10 percentile, the ability to predict it at an individual level, differs from between the 15th and the 100th percentile. So, just looking at that, if I was to tell you, you could get on a plane and it's got it's between the 15th 1.5 and the 100th percentile that it would crash, you probably wouldn't get in. So there's some technical aspects about why it would be difficult to use this at an individual level. And I think, Amy mentioned Jason Fletcher and others, and we have a paper coming out hopefully soon in Nature Genetics where we use sibling models and just say, these estimates that we have, it's a combination. It's genetic variation of these direct effects, it's demography, it's the population and assortative mating, and it's what you get from your relatives, so these indirect genetic effects as well. So, and then I guess looking at genetic correlations, so I've spoken about it a little bit more. Genetic correlations we often study. So we often look at a trait, let's say, such as educational attainment, and we see how genetically correlated is it with other traits. And, you'll see, for example, that what I study is timing of first birth, number of children, and the genetic correlation with that is extremely high with education and age at first birth. It's actually 0.74. So if you look at correlation, 0 means no correlation, 1 means a high correlation. So 0.74 is really high. That's a genetic correlation. We know from social sciences that, of course, you stay in school longer, you have children later, but this is a genetic correlation. So many sociogenomicists were, starting to realize and thinking, well, these genetic correlations are picking up a lot about the population and the environment. So, as a social scientist, I started to then look at the data as well and think, wow, there's a lot of sample selection. There's probably something biologically going on, what they call Pleiotropy. So one gene is associated with 2 or more traits. And all these correlations that we're picking up are genetic, but is it that the genetics that predict age at first birth and educational attainment, the correlation is 0.74. I'd be worried about applying that in a policy environment, because is it picking up fertility or is it picking up education? And I guess also we look a lot at gene environment interplay. So there's been a lot of studies by people like Jason Boardman, Jason Fletcher, Dalton Conley that have looked at these in detail. They look at policy changes and they see, for example, in relation to taxes and smoking, or in relation to changes in education, how that actually influences different groups using these scores. So there are some interesting applications, and we can focus on that later if you like. Some people, we look at gene environment interaction studies. So there are certain, mechanisms you can think about that some people who are highly religious or come from a household, a wealthier household, they have different opportunities and they have less exposure to certain detrimental traits or outcomes. And some of these things might be triggered by their context. You might have a predisposition for addiction, but it lies dormant because it hasn't been realized. So that's this gene environment interaction, which we study often. And then just to sort of conclude, as a demographer, I was really interested in the populations that have been studied. And so we have something in communications biology where we examined, the traits of the authors who look at these things, but also the population that have been studied. And we found, it was really striking to me as a social scientist that a lot of these genetic studies just combined 50 data sets together from different countries and age groups, and that would be very unusual for us to do. So we looked at this and we saw actually that in 2019, all genetic discoveries to date from these GWAS come from 72% come from 3 countries, the US, the UK, and Iceland. So think about that. This is where people are developing different, drug discoveries and many things. So this is, rooted in these populations. We also looked at them in terms of they were often older, they were more often female in the samples, and what does that mean? And I think Arbel's going to talk more about ancestry, and we also saw that you've heard it many times that it's overwhelmingly looking at ancestry. Now, whether that, or looking at European ancestry, now whether that's a helpful term, we'll, I'm sure, discuss. So I guess just to conclude, I've given some caveats to this. So, we've also studied, we saw that if you look at, we've examined our different genetic estimates across different countries and across different cohorts, and we found very many different estimates. So environment seems to matter, and environment also seems to be picked up in these genetic estimates that we're seeing. But despite all of that, it's an interesting frontier to look at, but I think that with, we're scientists, so we should always remain open to what we're finding. We should always remain open to a U-turn in our understanding. We should always, look for new techniques. And I think that's what we're doing at the moment. So it challenges our theoretical thinking. It allows us to ask new questions, and it allows us to get to those basics of nature and nurture, or nature versus nurture. So I think it's an interesting field for me to be in, and for many others at this moment, because it's quite exciting. And for me, it's not. I don't see it as problematic if some of our thoughts are overturned, but I do see it as problematic if we're thinking about applying these kind of traits when technically we haven't dug into what are we actually measuring. So what are these genetic correlations really measuring? So I'm going to turn it over to Arbel now, who will have a different perspective as well. So thank you.
Thanks, Melinda, and thank you, Amy. Hello, everyone. Before I sort of introduce my own research, I wanted to begin by saying how delighted I am to be part of this event, and thank the organizers for making it happen. I definitely share Amy's looking forward, but not really looking forward sense. And I will get more into that, but just knowing that all of you are here and seemingly are here about the numbers, that in itself shows that there's considerable interest in accurately and responsibly reporting this area of research and related LC aspects and consequences. So I'm a population geneticist at UT Austin. My research group studies how evolutionary processes such as natural selection shape genetic variation, and how genetic variation in turn maps to trait variation. So beyond basic science for science sake, I think a lot about how evolutionary population, statistical genetics can inform applications for human health and well-being. But up until a few years ago, I didn't really have any particular interest in GWAS, let alone GWAS for behavioral or social outcomes. In fact, GWAS seemed to me like this nightmare of data to be working with, because, essentially, as the name suggests, genome-wide association studies only tell us about correlations. And that correlation is not causation. Saying it is actually at the root of many of the issues that we're grappling with today. But this, series of research finding by myself and my colleagues really led GWAS to take this central stage in my research. And so I want to sort of tell you and give you a little bit of background about how this research about evolutionary questions sort of led my research to be intertwined with this age of GWAS, polygenic scores, and sociogenomics. So I will take this a step back because I don't want to assume that everyone knows exactly what we mean when we say GWAS and polygenic scores. So, so genome-wide association studies, just associate genotypes in various places in the genome, representing the entire genome. So they associate genetic variants with human traits, and perhaps one of the key findings that we have from a decade and a half of GWAS is that many traits that we care about, including behavioral traits, are almost always highly polygenic or complex, meaning that the bulk of heritable variation in the trait is due to not one or a few genes, but a combination of thousands of genetic variants of small individual effects. So why this is the case, the biology of this high polygenicity or complexity is in itself a fascinating question for another day. But for today, if we want to sort of like learn about the heritable variation contributing to high blood pressure, breast cancer, schizophrenia, and also, substance abuse or educational attainment, this observation of high polygenicity is really bad news, because what it means is that there is no single gene or 5 genes that have this large effect on a trait that we can understand the mechanism through or target an intervention through where interventions are relevant. But instead, there are this, tiny effects all over that are spread all over the genome, and it's incredibly difficult to pin down biological mechanisms when traits are so complex. But that is the reality. And yeah, so this sort of observation in itself shifted the focus of human geneticists such as myself from finding the gene for the trait to essentially summing over all of this tiny correlations to get meaningful predictions in the form of so-called polygenic scores. So, simply put, a polygenic score is exactly that. It's a polygenic score for a particular trait is generated by summing over the GWAS estimated effects of variants, and doing that for all of the variants that are carried by an individual to predict their trait value, be that their risk of schizophrenia or the years that they'll stay in school. And now getting to my own, background of coming to this from evolution. So years before any application of polygenic scores for clinical risk prediction, or the use was suggested for social science research or policy making with social and behavioral outcomes. Years before all of that, population geneticists such as myself have studied natural selection acting on complex traits by sort of detecting shifts in polygenic scores that happen over time or across populations. And the clearest example was of such an extreme shift that is consistent with natural selection was that of a high polygenic score across European subpopulations. And what my colleagues and I found is that our work sort of cast doubt into this previously ironclad example by showing that the signals were nearly absent when we were using this, newer, cleaner data from the UK Biobank. And the reason is that the effect estimates for genetic variants were biased due to population stratification. So the biases were usually very small, but when we sum over all of them and they act in some systematic way, it can lead to very wrong conclusions. So I would be happy to dig deeper into population stratification or population structure confounding a little later, and I think it should be of interest since the inconvenient truth is that we don't know how big of an effect or how big of a problem this is for a given GWAS this day and age. And there are good reasons to suspect that it's still a major factor that influences GWAS and polygenic scores for social and behavioral outcomes. So the bottom line, though, is that we showed that even tiny biases in effect size estimates can lead to this, usually wrong conclusion when they are summed up in a polygenic score. And this, this research finding is from only a couple of years ago, and it's looking at the traits that's supposedly very close to the middle biologically, much more so than behavioral or social traits that we think about as like very, very much mediated by the environment and social context. So, Hyde is like the poster child of polygenic trait research, and we thought that we had the best handle on this trait, and yet only 2 years ago we found that two of the largest studies the largest GWAS studies that were available that previously showed this results or led to this erroneous conclusions were that were also conducted by this large consortia. Two of this GWAS for Hyde suffered from systematic biases. So, while our initial focus was on this like relatively narrow point on the failure to reproduce evidence for natural selection acting on Hyde, our work made a much more general point about the sensitivity of polygenic scores to population structure, and also illustrated one reason why polygenic scores actually have limited portability across groups, and in this case across ancestry groups. So in in the context of today's discussion, let me just skip ahead and say that high prediction accuracy of, for example, a polygenic score for educational attainment is to an unknown extent could be due to the polygenic score just being highly genetic ancestry being highly correlated with things like income, as we most certainly know that it is. So this study was sort of the turning point of my research. I realized how far the science was from the proposed applications of polygenic score. And so this shifted my own attention from just the evolutionary applications to really thinking about how polygenic scores are being used and will be used in broader contexts. And it really also opened my eyes to the fact that that those problems are somewhat inherent in GWAS. So I also began to sort of realize how tempting it would be for non-experts to ignore this fact and this, technical sounding difficulties when they hear about a genetic predictor of a trait, and just think about this as a black box that represents some direct genetic effects of one's genotype on their phenotype. And sort of representing a propensity that maps to some biological mechanism that we don't understand, but that's what it represents. This is not the case, and to the extent that we don't know whether that's the case for a given polygenic score, and we should be more suspicious, I guess, when we're talking about traits that so heavily involve mediation, but by the environment. So. Since this work, we've also shown that even within homogeneous ancestry groups, the associations that we get between genetics and a trait can heavily depend on the environments, broadly defined, of the GWAS participants. So we found that the prediction accuracy of polygenic scores varies markedly depending on characteristics such as age, sex, socioeconomic status of the individuals, which are included in the GWAS, and the individuals for which the prediction is conducted. So all of this, goes to highlight somewhat underappreciated obstacles to the broad use of polygenic scores and to their interpretation. So today, GWAS and polygenic scores take this, central stage in my research, but this is really not because I'm now, suddenly a lover of correlations. It's mostly because I've sort of learned how important highlighting the windfalls, but also the pitfalls of this using GWAS and polygenic scores is. So this is a field where the applications are many steps ahead of where the science is, and in many cases, I believe that many steps ahead of what the science could even eventually, ideally, tell us. Okay, so yeah, I think at this point I can turn it over to Eric.
Thanks so much, Arbel. It is an honor to be with you and Melinda and Amy. I was asked to talk a little bit about my experience over the last couple of decades at the Hastings Center doing work in this space, that is the space of using genetics to try to understand complex behaviors. In the 2000s, I led a project called Wrestling with Behavioral Genetics, and it brought together enthusiasts about and critics of behavioral genetics to explore the scientific and the ethical territory. In 2004, just a year after the completion of the Human Genome Project, we produced a report and a book which described the state of the science. There was at that time some good news. Exploring genetic differences could, at least in principle, contribute to illuminating complex behaviors from autism and alcoholism. Given the twin adoption and family studies, it just wasn't any longer possible to deny that genes could help to explain observed or phenotypic differences. It's hard for some of us to remember how important it was to wrap our minds around the idea that it wasn't refrigerator mothers that were creating schizophrenia and autism. There was also, of course, a lot of lousy news on the science front at that time, as Dean Hamer announced in Science just a couple of years before we published our report. The results of the first molecular studies were at best disappointing and inconsistent. They just were not, as promised, turning up single genes with large effect sizes. In that piece in Science, Hamer mentioned 3 strategies that might move the field forward. Remember, this is 2004. 2 of those didn't pan out, but one of the ones he gestured at was GWAS, and as we've been hearing a lot about, that strategy is being taken very seriously if going even if it's going in for some rather heavy fire. In that 2004 report and book, I also tried to explain, I guess what's obvious now, which is that behavioral genetics touches on fundamental ethical ideas and values. Insofar as behavioral genetics is about behavior, it's always going to raise questions about freedom and responsibility. Just how free am I? To what extent are we learning anything about determinism? Of course, at this point, we know that genetic determinism is a dead end. Genes don't determine by themselves much of anything at all, not even PKU. And insofar as behavioral genetics is necessarily about genetic differences among us, research like this is always going to invite questions about equality. Just how equal are we? What's the relationship between moral equality and that other kind of descriptive equality that behavior geneticists are in fact on about. So to the extent that we had a conclusion in those years, it was the really remarkably novel one, I'm sure you will all agree, that this emerging science has potential for some very good uses and some very bad ones, and that it is incumbent upon those who do the research to communicate responsibly about what the science does and does not show. As Amy emphasized to start, that seems like still the order of the day. So today, I'm co-leading with Michelle Meyer at Geisinger a project called Wrestling with Social and Behavioral Genomics, aka Sociogenomics. And I want to underline that I am speaking today only for myself. The working group that I co-lead with Michelle is very much in the midst of wrestling with these matters. Like the Wrestling with Behavioral Genetics project, this one, Wrestling with Sociogenomics, also brings together enthusiasts about the science and critics of it to explore the scientific and the ethical territory. One thing that is new today relative to the earlier project is what we've been talking about, this GWAS technology that Hamer gestured at in 2002. As we've already heard, critics would say, and I think fairly, that GWAS are a very powerful street lamp, to refer to the old image of the drunk looking for his keys. It is really quite amazing to have the techno-scientific expertise to be able to identify correlations between single nucleotide polymorphisms and social outcomes. I don't mean by any stretch to dismiss the importance of that, but I do want to recognize that we are in the throes of, in the midst of, are using this remarkable technoscience that we may have exaggerated the importance of. Whether that's the case or not is yet to be seen, of course. Another thing that's new or different from then is, of course, that there's an even larger distance between the genes that are being investigated and the phenotypes that are being studied. It's one thing to study the relationship between genes and a rather close phenotype like autism, and it's much harder to get from genes to something like educational attainment, as Melinda and Arbel have emphasized. So given this distance, it is, as far as I can tell, going to mean that it's going to be really hard to come up with causal stories that start with genes and end up with such wildly complex phenotypes. And it seems to me worth remembering that some researchers don't need to be able to explain why what's going on is going on to be able to make predictions about future outcomes. Amazon doesn't understand why I like the things I seem to like, but it's sometimes depressingly good at making predictions about what I would like or how I will behave. And I would grant that we are still waiting for an excellent example of GWAS or polygenic scores being used for the sake of prediction, even in the context of the most simple medical conditions. Moreover, I would mention that even if polygenic scores aren't going to live up to the dream of contributing to causal explanations or even to predictions of social outcomes, they might, in fact, as I think Melinda was alluding to or referring to, they might well contribute to doing better social science by controlling for genetic differences. And intuitively, that ambition of using PGS to polygenic scores to control for the role of genetics and social outcomes makes a lot of intuitive sense to me. There again, I, for one, am waiting for an excellent example of sociogenomicists using polygenic scores to control for genetics in a way that not only enables them to do better social science, but leads to social interventions that are as genuinely useful as they hope they will be. Most importantly, the thing that's really new in our country today that wasn't the case when we were doing our behavioral genetics project is that we are in the midst of this reckoning with structural racism. The most fundamental ethical worries themselves about this kind of research aren't any different from the ones that we wrestled with back in the day. What does sociogenomics mean for free will and responsibility remains a question. There is interesting and important vignette-based research touching on the question of the impacts of old-fashioned genomic information about psychiatric conditions. As Matt Lebowitz and colleagues have pointed out, genetic information in the clinical context can or appear to, in some people, induce a kind of prognostic pessimism. That is, there's reason to believe that such reason could reduce some person's sense that they have a free enough will so that they can get better. And similarly, some vignette-based research by my colleague Lucas Matthew and colleagues, and his colleagues suggests that if there were genetic information about educational attainment, it could lead to students coming to believe that exerting their wills really couldn't do all that much to change their genetic destinies. All of that research, it seems to me, is very important, is ongoing, and we need to learn a lot more about. And I would recognize that results from genomic studies of this kind have been around for more than 50 years now. And I think it's hard for any of us to point to a kind of massive reduction in the attribution of personal or criminal responsibility in the light of these new genetic findings. Now, whether that could change in the future, I don't know. So finally, what about sociogenomics and equality? This does fill me and Amy and Arvel, and I wouldn't be surprised if it filled Linda too with some dread. Just talking about this can fill us with some hesitancy, if not dread. We do live in a society that's not only plagued by structural racism, but where many at the top of the income distribution believe that they are there because they deserve to be. Elizabeth Suhe, and forgive me if the pronunciation is wrong, a researcher at American University, Suhe and her colleagues recently reported in the Journal of Politics that the top 5% of Americans were more likely than others to emphasize intelligence and hard work as explanations for their success, and that the top 1% were unique in emphasizing both their genes and their choices as causes of those traits. That is, they emphasized genes and their choices, not social structures, as explaining why they were where they were. Now, the people that Suhe and colleagues surveyed were not relying on research from sociogenomics to justify their place in the social hierarchy, but they could in the future. And moreover, the racists have, as Amy Harmon has already written about, latched on to such research to advance the views they had long before sociogenomics was on the scene. But it is indeed new. It is a new weapon that they can use to their purposes, regardless of how poorly they understand what the research actually says. So at the moment, it might be fair to say that I worry less about the negative impact on the perceptions of freedom and responsibility than I once did, but I but my worry about equality is, if anything, grown. Does this mean I think sociogenomics research in general should be halted or banned? I'm open to the possibility that there are exceptions to the following rule, but as a rule of thumb, I don't think that bans are the way to go. For one thing, researchers have the right to investigate what they find inherently interesting. Thank God there are researchers like Robbie Widow who are asking out loud fundamental questions about whether there are certain kinds of analysis that ought not to be done. But for another reason that I worry about bans is that it's very hard to know how to implement them, given, as I think it was Amy who said, the data are hanging around, they're used in all sorts, for all sorts of medical research. It's very difficult to know how one would altogether ban research of this kind. So what that leaves somebody like me with is the exhortation to researchers and to, of course, journalists to communicate clearly about what the researchers are doing, why they're doing it, and what they have and haven't found. And as Amy said, it means that you journalists are going to have to try to push back when the people who write your headlines seek to grab eyeballs. I understand that you folks in the audience probably aren't writing the headlines that can do so much damage, but I think it's time for all of us to push back. The good news for people trying to communicate responsibly is that there have been initiatives ongoing. For at least a decade now, there's been a tradition of creating these frequently asked questions documented, frequently asked questions documents, or FAQs, which aim at explaining in layperson's terms what such research has and what research has not found. The Social Science Genomic Association Consortium in general, and Michelle Meyer, Patrick Turley, and Dan Benjamin in particular, can take pride in getting that tradition off the ground when they led the way to creating the first FAQ on the first educational attainment study that got so much attention. And another fantastic example of doing FAQs energetically and responsibly was created by the researchers at the Broad, including Robbie Widow, who did that study on same-sex sexual behavior. The further good news, and I'm about to wrap up, is that the working group that Michelle Meyer and I co-lead, or at least some subset of us, are planning to create a template for researchers who want to create FAQs for their studies. And I'll say that the last thing I'll say is that those researchers and all of you in the reading and all of you in the media who are interested can now go to a depository or repository of all of the FAQs that have been put together so far. That repository of FAQs was created by Daphne Marchenko and her colleagues. You can now find that repository on the Hastings Center website. Maybe Danielle can put up a link to help people get to it. But again, that is, I hope, a useful resource for journalists who want to communicate clearly about what such research has and has not found. Thank you. Over to you, Amy.
Thank you all so much. That was amazing, and I have a million questions. And I just want to encourage the audience to put questions in the Q&A, and no question is too basic. So please, I remember I was just thinking that like not even really understanding what educational attainment was. And actually, and one of my editors at the times when I was writing about it, I think it was in 2018, just, we had to like, he really didn't want me to use that phrase because it was so like, what do you mean? Do you mean achievement? Do you mean like years of, it was just, we tried to work out something that was a little more conversational. But so please do ask your questions in the Q&A. I want to just go back to Melinda. So Melinda, I want to be, I want to remind myself to ask you, I really want to know, and I think the audience will really be interested in learning about how your work has been reported on. I think you have examples, maybe not like the best example, an example of a story about how maybe it shouldn't be reported on. But before I ask you that, I just wanted to say, so Eric is saying he has not seen an example. So the great promise, right, of sociogenomics is, I think, is that it can help shape social policy. It can make research results on all sorts of interventions that are supposed to help society or parts of society be more accurate. So is there an example? I mean, what is the closest that the field has come, or or maybe about to approach, if you can give one, or maybe just talk a little bit about how that would work. Does that make sense?
Sorry, Amy, are you talking to Eric for the second
No,
half?
no, sorry, Melinda, I was talking to you. I was saying he was posing this question. He kind of said this offered this provocation that he said, this field is supposed to come up with, is supposed to, like, the ideal is for, I wrote it down, better social science by controlling for genetic differences, right? So social science would be improved and society would be improved if we can use results from these sociogenomic studies. But he said he hadn't seen a good example of that yet. And so I wondered if you might have one. Oh,
excuse me,
did I get that wrong?
I haven't seen a great example.
A great example. Okay, fair enough. Okay, well, Melinda, is there a good example or a great example or just any example that you might, I just wanted to sort of flesh out for people, what that might look like.
Yeah, so I'm not sure I want to get embroiled too much in using control variables. I'm not sure how interested this audience will be, but I might be surprised. But it's basically, it's talking about, okay, so you add a whole bunch of predictors and variables and you want to explain an outcome, like years of education. I agree. It's easier to understand. So years of education, or when somebody's going to have a baby, or levels of alcohol addiction or whatever. So you add a whole bunch of variables in a model. It's called multivariate. So often in the social sciences we had the usual suspects. So you would have your things such as age, sex, ethnicity, which you call race in the US, and all of these, where you come from, your family background, and all of these things. And, as I think it was, I read it in Paige Harden's book, those explain up to maybe 20%. That's your R squared of what you're looking at. The social ones, I'm just talking about the social ones. So, and then, you often you can add in these genetic predictors. So that's the idea. And I agree with Eric that, you can add these in and they can increase your explanation. But can they really do something? So, okay, I've increased my explanation. I now understand that when I add in a biological component, I increase my explanation. But I think when it's interesting is when you get into the gene environment studies, the gene environment correlation studies, because then you can really figure out. And, there's been some really interesting ones, particularly I mentioned it by Jason Fletcher and Jason Boardman, that looked at the differences of taxes on tobacco, for example, and how some people that were just very addicted, or, they had this genetic predisposition to being addicted to smoking, whatever tax increase you introduced, for example, that wouldn't influence that group. So I kind of think the discussion on control variables can get stalled, because then it's always about how much more can we predict in the model. And I don't think that really gets us to understanding, because we'll always get to the ceiling. And then I guess in relation to reporting, studying sexual behavior, timing of sexual behavior and children and fertility, I've been surprised. So, you always, and you saw it also with the same sex sexual behavior, regardless of all the great work they did, you saw that it was reported as, gene found or genes found for that to predict same-sex behavior. And for me, then you're put on this tabloid. In the UK, we have these tabloids. You're put on there as a Oxford professor predicts teenage sex or something like that. The genetic basis if there are all teenage pregnancies is genetic. Just those kind of things. And and I got a rash of mails, physically written mails and emails. Quite a few sex offenders, the way it was written up about you've explained everything to me now, and I understand why I do what I do. So this sort of what Eric was talking about, people ascribing some sort of reality or truth to these things. It wasn't even what we were looking at. I'm talking too much, Amy, but just, so I think thinking about controls is maybe a dead end in some ways. And thinking about responsible reporting is really important because it does touch people in different ways. And then, it comes back to the scientists often.
Okay, I just wanted to follow up for when I'm getting a lot of questions in the chat, which is the Q&A, which is great also. So, but on the but you mentioned the taxation, tobacco, people who are predisposed to be addicted to tobacco, and the implication of that. I just want to make sure this social policy implication is sort of explained, because I mean that seems like the most interesting, right? Or the idea, I mean, I think maybe in the future, if the the polygenics, depending on how robust the results are, this idea of educational interventions. I mean, there is this idea out there that you could have sort of precision education, or just like you could have precision medicine, or so. I mean, is the taxation for tobacco maybe the best example right now, or could you speak maybe a little bit more to?
Well, there's I'm sure that Arbel or others can speak to that as well. I mean, there's the with BMI and the FTO gene, and there's this there's some sort of studies are related to Alzheimer's or diseases. There's better relationships there, but I think in terms of addiction, you'll see probably, and I think Eric actually alluded to that, you'll see perhaps more applications policy related. Think about education, perhaps with dyslexia, being able to target dyslexic children earlier, for example, for interventions. But for the reasons I think that all three of us outlined in very different ways, we're not quite sure what's in the scores yet. And I'm working on this. And you said, Robbie Vado mentioned that, and others. So we're really trying to reckon with what we're measuring. And when I see that there's a 0.74 correlation with education and age at first birth, I can't believe that's entirely genetic. I just can't, and that's what we've said since about 2015 when we published this as well. So you have to be really you have to have some healthy skepticism about what you're measuring. So I wouldn't want that applied in a school, for that reason. I don't know what's in it.
Okay. Well, so on that note, I actually do want to go back to Arbel, because there are a few questions in the Q&A, and also I just wanted to make sure to give you a chance to sort of spell out this idea that I mentioned in my introduction, which is that there does seem to be discrepancies or different opinions, I guess, about how these scores can be applied to different populations. If there's sort of like a sharp divide, like, oh, there's this idea like, oh, we're only looking at people of European descent, so the scores are only relevant to people of European descent. I think that you were saying that that's not necessarily true, although it's also not necessarily true that the score is reliable for people within people of European descent. So if you can speak to that, I'm also just going to read you. Somebody was asking about the two GWAS studies that you found were flawed due to biases in the data. What traits were they studying? I just wanted clarification on that. Okay, yeah.
Yeah, so I guess I'll get to your first question first, and even before that, I wanted to follow up on some of the things that Eric mentioned. So you probably have to cut me off at some point, and I'll be very accepting of that. So, so I guess I wanted to follow up on the Eric's point on FAQs, frequently asked questions. I think it's a great it's a great culture to adopt, and we should all push ourselves to demand and provide those. Not only as a service to the public and to reporters that want to report this science, but it also, of course, forces the researchers to grapple more deeply with this questions. And I think the ones that Eric mentioned are good examples of that. However, I don't want the impression to be created that frequently asked questions means like right answers, or answers that are free from the biases of the researchers. And I also don't want the impression to be created, and I think it already has to some extent, that writing a detailed FAQ, means, some like legal release from responsibility or ethical release from responsibility for how you then independently communicate about the science or what you do in the paper. So, so just to take examples and again, without decreasing from the value of having the FAQs themselves, but the GWAS for sexual orientation. So writing a very detailed explanation on how the actual GWAS is done on customers of 23andMe that respond to this question of, I believe it was, have you ever had at least one sexual experience with the same-sex partner? And explaining, how this is very different, both on like doing this on 23andMe participants, looking at people that respond, like, the bias that you get by looking at people that responded to this question, the difference in this phenotype, people that answered this question, and people that are attracted to members of their own sex, the dichotomization of sexual orientation that sort of happens by dichotomizing the phenotype, all of that, acknowledging that in the FAQ should not mean that you're then, free to communicate the research again in the paper or by reporters later, or to reporters, as we found the genetic basis of sexual orientation.
I'm just going to interrupt you, Arbel, for a second, because I would like Eric to respond, because actually I was going to follow up on this idea also. So the FAQs, as a reporter, I found them very useful, and I would like to thank you for the authors. I know that people work really hard on those. But basically, I think Arbel is asking this question. You could also see them as just a more subtle form of PR for the paper. The researchers are asking the questions that, how do we ensure that the questions that the hardest questions are included in those FAQs? Are these FAQs enough? I think it's wonderful that there is a repository, because they are really helpful, and they spell out a lot of what's going on in these papers in lay language, and they do raise, some of the key questions, but you understand.
Excellent. So I as Arbel was speaking, this old acronym, "Oh God," came to mind. Remember, once upon a time, as you know, Amy, there were these geneticists who believed, one gene, one disease, right? We look back and we think, how could anybody have believed that simplistic a story? Well, today, I suppose, and always, right, there are people who imagine there's like this one ethical intervention, and it's going to make everything right. That's plainly silly, right? Of course, FAQs aren't going to fix things. They are one potentially very useful tool. And by the way, I thought Arbel's concerns or caveats about them were right on target. just because there's an FAQ doesn't mean that the answers offered are right or bias-free. And God knows it doesn't mean that anybody's released from the responsibility of trying to talk more broadly about these issues. And God knows the risk of commercialization is something to talk about for a long time. It's real, it's there, we should talk about it. Amy, I would push back on the PR bit. I mean, as skeptical and cynical as I am about pretty much everything, it's my impression that the people, well, the people who did the FAQs I'm talking about are genuinely trying to get the science and ethics right. And I don't think they imagine that they have, but I think they're trying. And by the way, in saying that, I'm not saying that, you know, good intentions are enough. As Catherine Bliss would remind us, they aren't. However, I would push back on the good PR point.
I mean, just I'm not meaning to impugn their motives, but they're okay. We'll leave it at that.
No, go ahead. Go ahead.
No, I mean, I just wonder if they could take submissions of questions, or if there could be there might be some way of making the FAQ process more open. But I feel like we're getting into the.
I would love to talk with you about that, because actually we are actively thinking about how to recommendations. So thank you. That's a wonderful idea.
Amy, can I pick up on commercialization?
Please.
Because I think we've been talking a lot about policy applications, and I think we should reflect on commercialization of what, so, you know, you could use these in policies. And I think it might not have escaped many people, but what happened with the same-sex sexual behavior, that an app was, you know, there was a product was released by a company saying, you know, we can do this with your genetic data to see how gay or lesbian you are, you know. So this is like really a problem. And I've had it with my own research looking at fertility and infertility, you know, contacted by companies saying, we'd like to use your polygenic score, you know, in relation in our fertility clinics for IVF treatments. So, you know, so you're imagining these vulnerable people coming in for fertility treatment and being sold something for $3,000, I think it was $3,000 to, you know, get their polygenic score on number of children ever born. So that's actually very, you know, it can get to quite dangerous applications and irresponsible ones.
I'm so glad you raised that.
Regulation in this area.
But can you spell out just what's, maybe it's obvious, but what is wrong with that? What is wrong with somebody,
so the
you know, using it, doing it
wrong with, yeah, so Arbel, maybe you want to weigh in, but I guess it's the same thing that would be wrong with using an educational years of education genetic score in a school. You know, so my polygenic score that I've looked at for number of children, so how many children you'll have and the timing of children, given, you know, that we don't, you know, we do these biological annotations and we find it's related to tissues, you know, in relation to, you know, sperm mobility, infertility, it's linked to endometriosis and things, but we actually, it's not just for sociogenomics, by the way, but we actually don't know the biological mechanisms for many of these, you know, variants that we find. So yeah, I think then by selling it, it's snake oil, you know, selling this to people, just to be really clear about it, because it won't, well, how would that help you when you're going for infertility treatment, you know, to know that you have a higher or lower polygenic score in relation to timing your number of children?
So we only have 8 minutes left. I want to go back to Arbel because you didn't get to answer my other question about the population, within population, between population, porting of the scores. If so, and then after Arbel answers that, maybe we can just all think about whether there's any last words you want to say on this.
Yeah, so I think, you know, in the, for example, in Paige Harmon's recent book, she describes, you know, the wonderful things that we can learn and the wonderful uses that we can have from GWAS and polygenic scores within population, but then says, you know, this says nothing about between population comparisons and, you know, touching on the FAQ that Eric mentioned from the Social Science Genetic Association Consortium. There's, they actually, in releasing this set of polygenic scores recently, they actually forbidden, make the user sign that they're not going to use it, not going to use these polygenic scores outside of European ancestry individuals, and are not going to make population comparisons. So, all of these examples are examples of people that I'm sure that have the best of intentions at heart, but I think this is incredibly fraught and misleading, and can also drive damage. First and foremost, because there's no such thing as a population. So there's people that have this incredibly complicated and family tree that joins them. So we all have this just continuum of relatedness that ties between us, and you can and I think this is like more of a point worth mentioning, worth making, that taking some discretization, so you can also just decide on a cutoff, an arbitrary cutoff of relatedness, where you will treat groups of genetic ancestry as independent labels. This is something that we often do in population genetics for essentially convenience of analysis. But those groups do not exist, but those distinctions do not exist in reality, and it's, and driving them through will only, and saying that there is such a thing as like, a European individual, or there are 5 ancestry labels, is in my mind just replacing race labels with a new word, and can contribute to essentialization. So, I don't think, I think it's sort of sending the wrong message to say that you can't use a polygenic score to predict the value on, on someone who's like beyond this line, in this arbitrary sense. The real, and we didn't really get to discuss the whole issue of polygenic score portability, the issue that the fact that polygenic scores tend to be much less predictive in samples that differ from the sample where the GWAS was performed. But the samples can be different from the GWAS sample in genetic ancestry. They can also differ from the GWAS sample in the environments that they experience, and social exposures, and other sorts of exposures. And we have no good sense at the moment of how each of these factors contributes to the portability problem. And this is a really central problem in the application of polygenic scores more broadly to benefit all of mankind, and in all of the traits that we're thinking about, not just of course social outcomes, but also biomedical outcomes. So I know I'm mentioning a lot of things, and we're nearing the end here, but just to get to that point, I think saying that polygenic scores and GWAS are capturing some causal direct effects on a trait, direct effects of one genotype on a phenotype when you're looking within population, but then don't do that when you're looking at cross populations, is just fraught scientifically from a few different reasons.
Okay. I mean, I feel like we need another panel just to talk about that issue. So maybe we can do that. But with the 3 minutes remaining, I guess I do want to ask you all to, if you have any quick concluding thoughts, and if not, we're good. There is a question in the Q&A that I'll read in case it helps to frame your thoughts, which is, should genomic data be used to shape social policies centered around inequality? What are the ethical implications of doing so? I feel like we need another panel for that as well, but that's out there. So Melinda, do you want to?
Okay, I don't think I'm going to answer that last question, but maybe just to sort of sort of summarize. I think that I think it's important to say, and just picking up on what Arbel said, I think a lot of us aren't arguing that this is causal, and we do a lot of downstream things to see, but it remains correlational. And I think that's just an important point. So that's not what sociogenomics is doing. And I really think that this understanding the difference of how you look at groups is kind of this difference between population geneticists and social scientists as well, that are used to looking at race, which is not a biological category. It's a politically, socially, culturally constructed category. And then we're dealing with, as you come in as a social scientist, you look at those ancestry categories. And the first thing you think of as a Canadian, the first thing I thought of was like, what about immigration and like all these mixed populations? The techniques don't even and they do now, they're starting to, counter that. But it doesn't fit, and it just repeating Arbel's point, it just doesn't fit into, our social constructs of how human populations are. And I'll stop now.
Eric, I'll give you the last word in our last minute here.
Well, I would just like to try to say where I stand. I'm really eager to try to find a middle way between despair about sociogenomic research, the idea that it's inherently evil and we're going to go to hell in a handbasket on the one hand, and on the other hand, what I take to be the kind of frankly extravagant hope and enthusiasm that one finds in this book that's been mentioned a couple of times. It's a very fine book, beautifully written by Paige Harden. I've tried to say why I don't buy her arguments in a post that I put on Bioethics Forum not long ago. I think it's worth trying to find a middle way between undue despair and undue hope. It's not easy, but that's what I'm trying to do.
That's a perfect last word. So thank you all for coming. I'm sorry we didn't get to all the questions in the Q&A, but we could talk about this for much longer. And I thank you to all the panelists. This is a fascinating conversation.
Thank you, Amy.
Bye.
Thank you. Bye.
Thank you all for joining us today, and a special thanks to Amy, Eric, Melinda, and Arbel. A recording of this discussion will be available shortly on ELSI Hub, as well as the Hastings Center website, thehastingscenter.org. And along with other resources, including the facts mentions, the FAQs mentioned, will be posted. Following today's event, you will receive an email with a survey about the event. Thank you so much for taking the time to complete this survey. Your answers will be used to help improve genomics and bioethics resources for journalists provided by the Hastings Center and the Center for ELSI Resources and Analysis. Please join us on October 26th for the next discussion in the series, Law Enforcement and Genetic Data with Sarah Zhang, a writer for The Atlantic, Ellen Wright-Clayton, a professor at Vanderbilt University, and Cece Moore, a genetic genealogist. We will notify you this week with registration information for the next event. Registration is restricted to journalists, journalism educators, and journalism students. Thanks again, and have a great rest of your day.